Hello students, welcome to the second lecture of the 4 lectures on glycogen, glycosaminoglycans and glycoconjugates. I'm not going to go over the objectives for this lecture because these objectives have been indicated in the first lecture. So you should listen to this lecture after you have listened to the first lecture. In this lecture, we will tackle the different glycoconjugates, look at the different classes, and also I have mentioned clinically relevant information related to some of the glycoconjugates. You have to remember this information because these are generally asked in the assessments. Before we classify the glycoconjugates, let us first try to understand what are glycosaminoglycans. Unfortunately, any biochemistry textbook assumes that students know what glycosaminoglycans are, and therefore they do not have a clear definition of this class of molecules. Now, glycosaminoglycans, often abbreviated as GAGs, also are referred to as mucopolysaccharides. So glycosaminoglycans and mucopolysaccharides are one and the same entity. Now, the GAG molecules are long unbranched polysaccharides containing a repeating disaccharide unit. And this particular aspect is shown here in this particular part of the slide. And you can see here it is consisting of a repeat of disaccharide units, and this is also shown here in the different types of GAGs that we will tackle over the next few slides. And you can see here they are consisting of a repeat of disaccharide units. Same here for chondroitin sulfate or dermatan sulfate or heparan sulfate or heparin. You can see they are consisting of a repeat of disaccharide units. The disaccharide units contain either of two modified sugars, N-acetyl galactosamine or N-acetyl glucosamine. We do not need to know the structure of these particular sugars, but we need to know which particular modified sugar is present in which of the GAGs, as we shall tackle the different GAGs in the next few slides. Apart from that, they contain uronic acid such as glucuronate or iduronate as part of their component. In general, glycosaminoglycans are highly negatively charged molecules with extended conformation that imparts high viscosity to the solution in which they reside. Primarily, glycosaminoglycans are present in the extracellular matrix. However, they are also found in secretory vesicles in some types of cells. Now we talk about the clinically relevant glycosaminoglycans. We start with hyaluronic acid. Hyaluronic acid, you need to remember this is the largest glycosaminoglycan. It binds large amounts of water to form viscous solutions and gels. Therefore, it is a major component of the synovial fluid. Glucuronic acid and N-acetyl glucosamine are the repeating disaccharides for this class of GAG, and it is the only non-sulfated glycosaminoglycan. This is also very, very important. So what I will ask you to do is to make note of this information separately so that you can quickly revise through these information prior to your assessment. The next type of clinically relevant GAG is heparin. Heparin is the natural anticoagulant that is present in our physiological milieu, and heparin prevents excessive fibrin formation during inflammatory or allergic reactions. Heparin is released by mast cells and basophils, and it enhances antithrombin 3 activity. We will look at the activity of antithrombin 3 when we go through the slides in lecture number 3. Antithrombin 3 activity leads to inactivation of activated serine protease coagulation factors such as factor 12, factor 11, factor 10, etc. You will be exposed to these coagulation factors when you take hematology. The repeating disaccharide in heparin is glucosamine and iduronic acid, and this particular information you also need to know. Next, we come to heparan sulfate. Heparan sulfate, it is located predominantly in the plasma membrane, where it imparts strong negative charge to the basement membrane. It has important function it is important for renal function because it repels albumin in the glomerular basement membrane. The repeating disaccharide in heparan sulfate is glucosamine and glucuronic acid, and you can see here that one of the components is similar to heparin. Therefore, I have written here you should compare the similarity with heparin. That will help you to remember this particular information. This is the only important glycosaminoglycan that is present in the central nervous system. That also is important for you to remember. Next, we move on to chondroitin sulfate. This is the most abundant glycosaminoglycan, and it is predominantly the glycosaminoglycan that is present in cartilage. In people who are suffering from osteoarthritis, they lose chondroitin sulfate because of digestion of the proteoglycans by metalloproteinases. Excess chondroitin sulfate and hyaluronic acid in the interstitial tissue is called myxedema, which is observed in Graves' disease, and this is also an important aspect that you should remember. The next important glycosaminoglycan is keratan sulfate, which is found in the cartilage. Lastly, we have dermatan sulfate, which is primarily found in skin, blood vessels, tendon, and valvular tissue in the heart. An increase in dermatan sulfate in the heart valves leads to myxomatous degeneration, which is associated with mitral valve prolapse. And this particular aspect is also important for you to remember for your assessment. Now we start classifying the 3 different types of glycoconjugates that occur in nature and that are predominantly present in our physiological milieu. The first glycoconjugate of importance are the proteoglycans. As you can see here in this particular part of the slide, this is how the schematic architecture of the proteoglycan looks like. These are macromolecules that are present predominantly on the cell surface or extracellular matrix, in which one or more sulfated glycosaminoglycan chains are covalently joined to a membrane protein or a secreted protein. So here you can see here, these are the sulfated glycosaminoglycan, heparan sulfate and chondroitin sulfate that is present, and the linkage happens through predominantly a serine residue, and these sulfated glycosaminoglycans, as indicated here in this part of the slide, are linked to a membrane protein, and this is the extracellular part of the protein, and this is the transmembrane part, which already we looked at when we studied amino acids. So you know what a transmembrane region of a protein looks like, and this is the intracellular part. Proteoglycans are major components of all electrocellular matrices. And you can see here, this is the integrin protein to which the proteoglycans are attached. So as you can see, if I translate this particular schematic to this particular figure, you can see here integrin is a transmembrane protein, and to the extracellular domain of which the proteoglycans are attached. Next, we come to the second class of glycoconjugates, which are known as glycoproteins. Glycoproteins have one or several oligosaccharides of varying complexity joined covalently to a protein. And as you can see here, this is a membrane protein. Again, this is the extracellular part, this is the transmembrane part, and this is the intracellular part. To the extracellular part of this protein, you can see that oligosaccharides are attached, and you can see here there are two types of linkages, and we will talk about these two types of linkages in the third lecture. The first is called the N-glycosidic linkage, which happens between the oligosaccharide and asparagine residue, whereas the O-glycosidic linkage happens between serine and threonine residues and the oligosaccharide. Now these kind of glycoproteins or these classes of glycoconjugates are usually found on the outer face of the plasma membrane, in the extracellular matrix, or in the blood. The oligosaccharide portions of glycoproteins are very heterogeneous and are rich in structural information. Therefore, they help clinicians or as well as researchers in tissue typing. One of the important aspects that you need to know is that some cytosolic and nuclear proteins can be glycosylated as well. However, the take-home message here is glycoproteins are oligosaccharides that are linked to proteins where the linkage happens through 2 or 3 specific amino acid residues, where N-glycosidic linkage happens between the oligosaccharides and the asparagine residue, whereas the O-glycosidic linkage happens between the serine and threonine residue and the oligosaccharide. These are some of the important functions of glycoproteins. I have tabulated them for you. I'm not going to read through them, and as you can see here, they have a diverse function inside our body, and as you go through the medical program, you will be exposed to several of these glycoproteins in specific, where their functions will be discussed and elaborated in detail. Again, I come back to a slide where I have highlighted some of the important clinical functions of glycoproteins. First, we have the blood group antigens, which are present on the red blood cells, and this is part of a reading material that has been provided to you at the end of the slide set that has been uploaded on the learning management system. Further, they are also present on circulating blood proteins such as coagulation factors and tumor markers such as prostate-specific antigen. Next, we have laminin, which is an important glycoprotein present in the basement membrane, which maintains integrity of the basement membrane. You have to remember this, that it also binds to integrin receptors, producing different types of physiological effect, but most importantly, it produces a growth factor effect. Fibronectin is an adhesive glycoprotein which binds to cell surface receptors, including the integrin receptors, as with laminin, and attaches cells to the extracellular matrix. Glycoproteins are also important components of lysosomal enzymes, and we have discussed this later in the 4th lecture. The third class of glycoconjugates are the glycosphingolipids. which are shown here in this part of the slide. Glycosphingolipids are plasma membrane lipids in which the hydrophilic head groups are oligosaccharides. Both glycoproteins and glycosphingolipids are recognized and bound by carbohydrate binding proteins called lectins, and you can see here in this particular part of the slide. So let's look at this part of the slide. These are the oligosaccharides, which are the hydrophilic part of glycosphingolipids. The aspect of sphingolipid we will talk about in next week's lecture when we talk about complex lipids. This oligosaccharide that is bound to the membrane lipid is hydrophilic, and they are important binding sites for a group of proteins called lectins, and you can see this in this particular part of the slide where the lectin is binding to the oligosaccharide that is bound to the membrane protein. So that's it. So in this particular lecture, we have narrated what are glycosaminoglycans. I have identified some of the specific clinical information in relation to glycosaminoglycans that you need to remember. Please take a note of that because that will be asked in the assessments and also are inquired in the boards. We have also classified glycoconjugates according to their structural orientation into 3 classes, and we have looked at the basic aspects of these 3 classes. We will need to focus on the specific structural aspects of some of these glycoconjugates, which we shall tackle in the 3rd lecture. With that, I end this lecture. Thank you for listening.